The purpose of this video lecture is to re-emphasize some of the material that you read about this week in the Citable.com article and also in the Concepts in Biology book. And specifically, the focus here is on plant population ecology. So both of those readings were pretty general. They were talking about the dynamics and how we study both plant and animal populations. In this particular lecture, I'm going to be trying to use some local plant examples to re-emphasize some of those concepts. First of all, I want to make sure everybody knows what a population is. It's a group of individuals of the same species living in a defined area. The plant pictured here is the Wairika flax, and we have several populations of this local endemic and endangered plant around Wairika. So population ecologists are people who analyze factors that affect population size and how and why it changes through time. So they are really interested in small populations like the Wairika flax. They want to know what's going on with this population. Why is it small? Are there outside factors that are limiting its growth? Are there internal factors? Is it not reproducing and producing viable seeds? What's going on that's caused this population to become small, and are there things we can do to try to help it recover? So those are the kinds of things that population ecologists are interested in. So you read a little bit in the Citable.com article about this really big question in ecology. What determines the distribution and abundance of organisms? Or, what determines where we find a particular organism living? For example, why do we only find the Wairika flax growing in this little area around Wairika and nowhere else on Earth? A lot of times, the limitations of a species distribution are a result of non-living factors in their environment. And this is the kind of stuff that we've been talking about for the last couple weeks as we were examining the impacts of climate and soils on plant growth. Those are non-living factors that are really important in determining where specific plant species can grow. So I have some of those things listed here. The temperature, the amount of water would relate to what we were talking about last week with climate, also the amount of sunlight, and then things like salinity and soil were the kinds of things that we were talking about a couple weeks ago when we were talking about soils and plant growth. There's one more thing on here, a really important non-living factor that has a big impact on determining where a particular organism can live, and that's disturbance. So to an ecologist, disturbance is a discrete event that will decrease the biomass, the plant biomass of a community. So these are things like fire or some kind of hurricane or storm or something like that that will come in and knock out certain organisms in the community. And these are all examples of non-living things that, organisms have a range of all of these things that they can tolerate, and they're only living in places on our planet where all of these different non-living factors are tolerable for them at the same time. We layer on top of that biotic factors. So the interactions that organisms are having with other living things can also determine where we find them living on our globe. We're going to talk a lot more about this next week, but for now I just want you to be familiar with the concept of a mutualism. certain plants have very specific relationships with specific pollinators. Those pollinators are helping them reproduce. The plants in return are often feeding the pollinators. But if the plant is relying on a single pollinator, we won't find that plant living in places where that pollinator is absent. The same can be true of herbivores. We see some plants, only living in a forest understory in places where there's a lot of herbivores out in the open. So it can determine, herbivory can determine where we're finding certain organisms, and so can competition. These are interactions between plants or other organisms that limit their survival and limit their growth, usually because they're depleting each other's limiting resources. So with plants, they can have competition for nutrients or competition for light, some of those essential things that they need to get out of a habitat in order to survive. If the competition is too strong, they might not be able to live in that particular place. So this is kind of the first layer of determining, why we see a certain species living in a certain place, the abiotic factors and then the biotic factors. Okay, so now I want to talk a little bit about how a population ecologist might study a population. First of all, it's a little bit confusing, the concept of a population boundary, because a lot of times there are not natural boundaries for a population in nature. So the person studying the population a lot of times is determining the boundary. It can be pretty easy to determine a boundary. For example, if the population of interest is on an island, or if you work for a county agency or a state agency, those political boundaries are probably going to become the boundaries of your population of interest. But usually the ecologist is determining what are the boundaries around the population. It could be determined by geographical features like a mountain range or something like that. So oftentimes within that boundary, the ecologist wants to know the number of individuals that exist in the population. And that can be done a couple of ways. For plants, we can count them, and that might be a viable option for really small populations like we have with the Wairika flax. But for really large populations like we see here in the picture of California poppy or the yellow-flowered plant here, which is in the genus Lasthania, it might be necessary to census a subplot to determine the number of individuals. So that would entail having a meter square plot or half a meter square plot that you put down on the ground in various locations within your population boundary, and you count the number of individuals just within that subplot. And you do that a few times in order to come up with an average number of individuals per meter squared, which would be a density, or you can actually just take that number on a per meter squared and you can multiply it by the size of your population boundary, and then you can get an estimate of the number of individuals in that population. Next, let's take a look at another population attribute called distribution or dispersion. You saw this in the Concepts in Biology reading, but now I want to give you some examples of dispersion and some of the mechanisms that might be driving it in plant populations. First of all, recall that there are 3 different options for the dispersion or distribution of a population. It could be uniform, like we see here. It could be random, like we see in this image, or it could be clumped, like we see in the bottom box. Uniform distribution can be caused by negative interactions between individuals. There are a couple of examples of this from the plant kingdom, including some local plants. For example, this species, Salvia leucophylla, has been described as having uniform distribution as a result of chemicals that it exudes from its roots. Those chemicals prevent other plants from establishing nearby, causing the populations to have this uniform distribution. We see the same thing in creosote populations, which you can see down here. It's this shrub that grows in really arid environments in California and other places in the Southwest. Creosote has been described as having uniform distribution as well. It's not a result of allelopathy or the production of toxic chemicals like in salvia, but rather the result of really strong competition between individuals. So there's such limited amounts of water and nutrients in these arid soils that it's really difficult for two creosote plants to grow in close proximity to each other. They compete too strongly for limiting resources, and as a result, the ones that survive tend to be pretty far from each other, and we see this uniform distribution as a result of that strong competition. I'm going to talk about this bottom box, and then we'll come back to the example of random distribution in dandelion. So clumped distribution can result from a bunch of different factors. It could be caused by positive interactions between organisms, so that's when they're actually helping each other out. I saw some examples of this when I was doing salt marsh restoration work. We tended to see new recruitment of seedlings near plants that were already established. So we were seeing a clumped distribution in these communities that we were trying to establish. The reason was that where there were already plants in place, the salt marsh plain was less salty, so the plants that were there were shading the marsh plain, making it less salty and more conducive to recruitment of seedlings. We also sometimes see this clumped distribution in plants that have limited seed dispersal, so that would be plants that generate really heavy seeds that don't tend to move very far from the mother plant, so they'll end up with clump distribution as well. Clump distribution can also result from the heterogeneous locations of the resources in the environment. So plant might require or will require a certain suite of resources, and they may not be uniformly distributed in the environment. So the habitat might only be suitable in small sections within the landscape. So as a result, we might see clump distribution. An example that I have of that is the Wairika phlox. So the Wairika phlox is only found in scattered populations that are clumped together in certain areas around Wairika. It seems to be associated with serpentine soil, so it's only going to be able to establish in habitats that meet those particular requirements, that have those kinds of soils. And those soils aren't going to be uniformly distributed in the landscape. Okay, finally, let's take a look at our example here of random distribution. So dandelions are thought to have random distribution in a lawn. Unlike the plants that have really heavy seeds, dandelions make these light wind-blown seeds, and they can just kind of scatter anywhere throughout the lawn. And a lawn is going to be a pretty homogeneous environment. And if a seed can survive and establish in one place in the lawn, it's likely that it's going to be able to do so anywhere. So those kinds of factors could lead to a random distribution of plants in the environment. Okay, now let's switch gears and talk about population growth. So we might be interested in the size of the population, the density of the population, the distribution of the individuals within our population, or we might be interested in knowing, is that population growing, stable, or is it declining? One thing that we sometimes see with natural populations is exponential growth. And this is growth in which the rate of growth is accelerating. So the rate of growth here can be seen when we look at the slope of the line on this curve. So down here, the slope is pretty level, pretty close to zero. But as we move over through time, we see that that slope is increasing. It's getting steeper. This happens in natural populations because as there's more individuals out there in the population, they all have the ability to reproduce themselves. So as the population gets bigger, the growth rate will increase. This cannot be sustained in nature. Eventually, something is going to happen that causes the population growth to stop. But we do see it for periods of time in certain populations. For example, we see this in some invasive species, like the one that's pictured here. This is common in Siskiyou County. It's called Marlham mustard or Dyer's woad. And it was brought over to California. At one point, it wasn't here at all, and now we see certain areas like out in the Scott Valley where there are huge populations. It's undergone exponential growth. And we commonly hear ecologists who study invasive plants talking about exponential growth. So population ecologists aren't only interested in endangered populations, small populations, they're also interested in invasive plant populations because often they're trying to control these populations. Okay, so exponential growth is the growth of a population where the growth rate over time is increasing. We know that that kind of growth can't be sustained. Eventually, what we see is that growth rate declining back to a horizontal line, which means no growth, which means a growth rate of zero. This model of population growth, where we get these S-shaped curves, is referred to as logistic growth. So it's growth of a population that accelerates initially at a growing rate, but then slows to zero. So this typically happens, the growth rate will slow to zero around what's called the carrying capacity for that particular population in that particular location. So the carrying capacity, which we've seen shown here as this dotted line, it's the population size represented by this dotted line on this graph. Ecologists tend to use the letter K to denote carrying capacity, and it is defined as the maximum population size of a species that can be sustained by a given geographic area. So the Dyer's woad is a terrible weed, and it's exponentially increased its populations, but even the Dyer's woad eventually will level off and hit a carrying capacity where the growth rate will no longer be positive. It'll slow to zero. We might not be willing to let that happen because some of these invasive plants have such detrimental effects on our native communities or on our agricultural economies, and in which case we'll try to lower that population size through removal efforts. Okay, so this slide tells us some of the things that might be happening that cause a population's growth rate to decline to zero when it reaches its carrying capacity. So these are like natural populations, not managed populations like some of our weed populations. At carrying capacity, when the population is really high, the individuals are going to start to compete more for limited resources. That might slow the growth rate. There could be diseases that start to spread, fungal diseases or viral diseases that start to spread through plant populations. Herbivores might start attacking the plants, or allelopathy, which is when chemicals are released in the root zone by plants that could start to limit the growth of a population once the population hits carrying capacity. It's at a really high level at that point. There may be factors that are affecting the growth rate of a population that have nothing to do with the density of individuals. And these ecologists refer to as density independent factors. These are things that are going to decrease the number of individuals in a population regardless of how many are present. So this could be things like disturbances or pollution or unusual weather patterns. Other factors that are intrinsic to the organism, called life history strategies, can also have a big impact on population dynamics. Life history strategies include traits that affect an organism's schedule of reproduction and their survival. So, for example, some plants reproduce really quickly, whereas others may take decades to reproduce. Some of them, produce offspring that have really high survival rates. Some of them produce many offspring that have really low survival rates. All of these traits taken together would be referred to as the organism's life history strategy. These strategies come about because energy is limited. So one organism can't do it all. It can't devote a lot of energy to its own body growing really large and also have a bunch of offspring and provide a lot of resources for those offspring. There's just not enough energy. That organism doesn't have enough energy to make all of that happen. So these life history strategies often involve trade-offs. Some annual plants represent sort of an extreme case of life history strategies. They're over here on this side of the continuum. These are plants that are generally small in stature, so they're not devoting a lot of energy to their own growth. Instead, they're reproducing really fast, but they're only reproducing once. So this is called semelparity, when organisms just produce once and then they die after reproduction. They can produce a lot of seeds. So some of these annual plants produce hundreds of thousands of seeds. This is the annual plant Salicornia bigelovii that I worked on in graduate school. And each one of these individuals can produce hundreds of thousands of seeds. They don't get really big. They're maybe about 25 centimeters tall. But a huge portion of their biomass is devoted to reproduction. This strategy, with emphasizing reproduction, getting it over with fast, is favored by plants that live in unstable environments. In an unstable environment, you never know when the next flood is going to come, when the entire population is going to get covered by erosion, and the habitat's no longer going to be suitable for that particular organism. So the strategy allows these plants, when the conditions are favorable, to really quickly ramp up their populations through all of this reproduction. On the other end of the continuum, we have some plants that have a really different life history strategy. So really long-lived perennial plants like trees. Obviously, these are large organisms, so they devote a lot of their resources to growing their bodies. They can be huge. We often see in these long-lived perennials that reproduction is occurring more than once. We call that iteroparity. It may be delayed, so the organism may spend decades just growing in the understory before it even starts to reproduce. In some cases, we see these long-lived perennials producing larger seeds, fewer of them, and larger seeds. This is kind of the plant equivalent of high parental care. So that seed is a little packet of nutritive tissue, and then their offspring, their embryo, is tucked inside a little seed coat. So in a larger seed, there's usually more nutritive tissue that's going to give their offspring an advantage in a really competitive environment. And oftentimes we see trees and forests growing in high competition, stable environments. This is like prime real estate. So this life history strategy is a lot more conservative, and it doesn't emphasize reproduction as much. And it sort of would be what we would expect in populations that are growing at their carrying capacity rather than ramping up their populations like we saw in the annual plant example. Wilson and MacArthur talked about these two extremes, the annual plant and the perennial plant that we just looked at, using the terms R selected versus K selected. And you read a little bit about this in Concepts in Biology. R refers to the per capita rate of increase, so that's the growth rate of the curve, of the population growth curve, and it's going to be really high in R selected species. Like we just said, this kind of strategy is favored in new or disturbed environments where there's less competition, and generally there's little parental care. In the plant world, that means small seeds. On the other extreme, we saw the perennial plant life history strategy, which Wilson and MacArthur referred to as K-selected. Those are organisms that we would expect to be in stable populations, living at their carrying capacity. So K refers to the carrying capacity. These life history strategies favor efficient use of resources, and it's going to be favored in a highly competitive environment. Okay, so in summary, these tools of population ecology allow us to determine if a population is shrinking, stable, or growing. It also allows us to try to understand the extrinsic factors that may be causing those dynamics. And population ecology also gives us some tools for looking at the intrinsic factors about the population, the life history strategies that might help us determine, why we're seeing the patterns that we're seeing out there. Hopefully this lecture helps you understand some of the material that you read this week a little bit better. Let me know if you have any questions in this week's discussion.